Hi guys, how are you? Hope you're good. Ready for chapter 8? We're gonna talk about microbial genetics today. I love this chapter. It's a very fun chapter to learn, I think. Maybe because um, I've been working with molecular biology and molecular sciences for a long time, so I just find all this super amusing. So let's start with our learning objectives. Um, so we have here definition of gene, chromosome, DNA composition, um, which is what is the nucleic acid, why DNA is a double helix, which are the four nitrogenous bases. So we're going to talk about all of that in a minute and where you're going to find this. Definition of genome, genotype versus phenotype. What is the gene regulation by an operon? We're going to talk about that. Mechanisms of genetic change in bacteria. We're going to talk about the horizontal and the vertical type of genetic change. What's a plasmid, which is kind of included in this topic, and general steps for DNA duplication, transcription, and translation of proteins. So, for this last topic here, for this topic over here, and this topic over here, a lot of it, including DNA composition mostly, and all that is described in this last one, are going to be separate videos that are included in the same playlist. So after you watch this video, make sure you go to the next ones to watch these other topics, okay? And why do I put them as separate topics? These are things that you were supposed to learn or know from A&P. So I always assume that you took A&P before in micro. That's what we always assume, right? All right, so let's talk about this. We have, let's see if I can move my face a little bit here. We have some definitions to work with here, which are genetics, which is the study of genes. So we're good here, right? How genes carry information, how information is expressed, and how genes are replicated. So how genes are replicated is like how one strand of DNA makes another strand of DNA. How information is expressed means how DNA makes RNA that makes proteins. All that is explained in the separate videos that I'm mentioning to you in the same playlist. Chromosomes. What is a chromosome? This is a chromosome. This is actually already duplicated chromosome. When the chromosome is not duplicated, it just looks like one little strand here. But when it's duplicated, you have one here and one here, right? Cool. What is the chromosome made of? The material that coils up here to make the chromosome is called chromatin. Okay, so it's all coiled, and it's coiled because when the cell is dividing, that's the form that you find the chromosome. Think about you going on a trip and you have to pack all your stuff to go on the trip. So if your belongings are not packed, what's gonna happen? You're gonna start losing them along the way, right? So that's why you need to pack them. Same thing here. So when you see chromatin packed, like coiled up like this, it's very likely because the cell is ready to divide. So the migration of these chromosomes to the two poles of the cell and mitosis and everything that we learned before will happen with the material packed. What is the chromatin made of? If you get one piece of this little strand and you uncoil it, what is that? That uncoil is the DNA. I'm not going to be able to draw DNA here with this pad, but let's try. The DNA, right? Okay, so chromatin is made of DNA. Chromosome is made of chromatin. Got it? Okay, so now let's talk about what is a gene. Is a gene this whole thing, one whole thing here is one gene? No. A gene, let me get another color that would make it easier to see. A gene would be this here. One little piece, so this is a gene, and here is the same gene on the other chromatin, which is, they're probably going to be identical genes or very similar genes, and they're always going to code for the same trait. Okay, so if I'm talking about eye color, for example, which is the typical example, right? Then you know that this gene here and this gene here both code for eye color. So what is a gene? It's a segment of the DNA that encodes functional products, which are proteins. So one gene needs to encode a whole protein. You don't have a protein that is made of one piece of gene here and one piece of gene there. A whole gene makes one protein, okay? Genome is the combination of all the chromosomes, which include all your genes in your body or in an organism's body, right? And what do I have here? What is this? This is what we call karyotype. What is the karyotype? It is all the chromosomes that are present in some organism's body. So how many pairs do we have? We do have 23, right? This is probably another organism. It's probably another or eukaryote, because how do I know? Because remember, bacteria has only one chromosome, sometimes more than one, but most of the time one, and it's circular. So when I see chromosomes that are linear, like this, I know that this is not a prokaryote, okay? So in this case, let's say this is the karyotype of an eukaryote. Is this the organism's genome? The genes, all the genetic information present in all these chromosomes? Yes, it is, okay? And we call them pairs because you find chromosomes that are practically identical, and then that's pair, for example, pair 12, right? Pair 11, pair 10. Cool, so we have 23 pairs, 46 chromosomes. Bacteria, only one chromosome that is circular and is not even paired, okay? The genetic code is a set of rules that determine how a nucleotide sequence is converted to an amino acid sequence of a protein. So what is the central dogma of our genetic code? The DNA, which is in your nucleus, and in the bacterium, nucleoid, by, what's the process here? Let's write sideways. By translation. No, all wrong. Bad me. By transcription. Oops. Back to my pen. By transcription, we'll make RNA. Which RNA? The messenger RNA. It's always going to do the transfer RNA. And also going to do some other regulatory RNAs that participate in the process that we're not going to mention here. And that transcription and mRNA will be, now yes, translation to proteins. What is the translation? Is translation what we call expression? Yes. Expression of a gene is the fact that that gene can make through the RNA a protein. Cool? Great. Now let's talk about what is genotype and what is phenotype. So genotype is the fact that you have those genes in your genome. So This is one gene. You see this little green stripe. This is another gene. That's the allele for that green gene. This is the allele for the blue gene, right? So this is a pair of chromosomes. You see, I'm talking about an animal, an insect, not a bacterium in this case. But for the bacterium, it would be the same thing. You have that circular chromosome, and you were going to have a gene here and a gene here, one that codes for something related to, I don't know, carbohydrate synthesis, some enzyme, the other one that codes for proteins that are involved in some housekeeping function, and so on. What is the phenotype? It is, whoops, it is the expression as proteins, right? Expression of these genes. That's the phenotype. So phenotype is how you look, and genotype is the type of genes you have that confer that look. Okay? Like I said, bacteria usually have a single circular chromosome made of DNA and associated proteins. That are proteins that coil where the DNA coils around, okay, to just keep that material, genetic material packed. This is the chromosome of a bacterium, and this image is just, it's a transmission electron microscopy image just to show you how extensive is that circle of DNA inside a bacterium, but it fits inside a bacterial cell, which is this. Okay? Cool. So now we're going to talk about vertical versus horizontal transfer. If I have one bacterium here, and another here, and another here, and they're all going to be mummies. So they're all going to divide by binary fission into two bacteria, right? Same thing here, same thing here. Okay. The genetic, so now let's make it in purple. The genetic material that is, whoops, this is not going to work. Hold on. The genetic material that is inside this bacterium before this bacterium divides is going to be here. This is the DNA, right? Do you agree that this DNA is gonna first duplicate in two, so you can give one to each daughter cell during the binary fission process? Okay. Do you agree that whatever genes this mommy bacteria cell has, the daughter cells are gonna have it identical? Cool. So if we all agree with that, we are talking about vertical transfer of genes. Okay? Mom gives to the daughters, done. Is there a dad here? No bacterial dad. Okay? So, still talking about the flow of genetic information through vertical gene transfer, look at this. DNA inside a parent cell. They don't want to call mommy because I'm being kind of feministic here, say mommy, but it could be just a general bacterial parent, okay? So that bacterial parent is going to transcript an RNA and translate a protein. Transcribe an RNA and translate a protein. That's expression. What another concept that is related to the flow of genetic material? Recombination. You can get a little piece of genetic material here. You see how it was like a light purple? That comes from somewhere else, and we're gonna learn that. And this is usually what we call horizontal in some cases, but it could also just be a recombination within the same cell. And replication when the genetic material divides. Okay, so we know all this. Let's just move on. Now, we need to be a little more sharp to understand this. So there is a control of the expression of proteins that happens before the translation and even before the transcription. There is a type of regulation that happens in the DNA, on the DNA level. What is that? It could be repression and it could be induction. So what is repression? Repression happens to genes that are what we call constitutive. Constitutive genes are genes that are always being translated, and they do not stop that translation into proteins until there is a repressor to stop it. So the break is an actual break. You need to break it, and if you don't stop it, it's just going to keep going and producing that protein, and that could not be very effective, right? In terms of using a lot of energy for something that you may not need. Okay? Now, let's talk about induction. Induction is the other way. That gene is usually with the break on, so there's something before that gene that makes that DNA, RNA polymerase that would make the RNA to make the protein later stop, and you need to remove that repressor in order for that gene to be expressed. Okay? If you didn't get it, please go back and hear what I said again. Okay? So, we are going to focus more on induction, which uses an inducer. Okay? I'm going to give you the main characters from this story, and then I'm going to play a little movie for you. Okay? So this is a piece of the chromosome of the bacterium. Okay? So here we're talking about an operon that is called lac operon. What is the lac operon? Operon is a region of the DNA that includes the promoter, the operator, and the genes. Which are these genes? In our case, these genes code for proteins that are enzymes. So all the Z, Y, and A are enzymes. Oops. That digest or break down, whatever you want to say, digest lactose. Okay? So these genes are usually not expressed. Why? So then that's what we're going to talk about now. Second part, not expressed because there is a protein that is made by this regulatory gene. This is the regulatory gene before the promoter, right? And that gene is usually attached to the operator. So the repressor protein is made by the regulatory gene and attaches to the operator. What happens next? Well, that's RNA polymerase, the enzyme that would make Z, Y, and A produce mRNAs for these three genes, right? And then with these mRNAs, consequently, you would have these three proteins coded by Z, Y, and A. Which proteins are these? Enzymes that digest lactose. When does a bacterium need enzymes that digest lactose? When lactose is present. Do they need this when lactose is not present? No. Why are they going to make all this stuff that they don't need? Right? It's just useless energy. It's like you're using energy just to waste it, right? Okay, so how do you make this repressor to detach or not to attach? It's actually to detach from the operator, so you can transcribe those RNAs for Z, Y, and A? Well, remember I said you need to have lactose present to make those enzymes so they can digest lactose and the bacterial cell can use that lactose? There we go. Allolactose is the lactose, that is a form of lactose, that binds to the repressor. When it binds to the repressor, what happens here? The operator is free. And if the operator is free, what's going to happen to my RNA polymerase that is here? It's going to slide through and move along and make these 3 RNAs, make the 3 proteins that are called beta-lactosidase, permease, and transacetylase, and do what in the end? Digest lactose. Got it? Okay. You may say, this is complicated. I don't understand why I need to know this. And I'm going to tell you why you need to know this. You're like, but I don't know. I think I'm going to be a dental hygienist or I want to be a nurse. Why do I need to know all these gene regulations? So listen to this. If you put your brain to work to understand something from this level of complexity that may sound complex to you, what's going to happen? You're wiring your brain to learn how to teach yourself new things. So I'm not just teaching you what's an inducible operon here. I'm teaching you how to use your brain to do more complex tasks. Because once your brain gets wired into that type of function, which is more complex, you're in business. Anything that is complex, that is presented to you, you already wire your brain to do that by learning what's an inducible operon. So I'm just helping you here, not just to understand this, but it's like to wire your brain for future complex tasks. Make sense? So don't be mad at me that I want you to know this. So now I'm going to show you a video that talks about this. Let me move. So this video is actually from, make it bigger for you. This video is from your Mastering Microbiology page. If you don't have Mastering Microbiology, you may not be able to see it, but I'm showing it to you right now, okay? So let's play it. I'm just gonna be quiet for a minute so you can watch it. I'm gonna make the speed a little higher so you can just see. This is exactly what I just told you, okay? Promoter, regulatory gene. This is gonna make the repressor. Repressor is this part. It's gonna bind to the operator. So
includes a promoter, an operator, and 3 structural genes encoding proteins involved in the catabolism of lactose. The lac operon is an inducible operon, meaning that the genes are usually not transcribed. When lactose is available to the cell, the operon is induced or stimulated to produce the proteins of lactose catabolism. The E. coli chromosome contains a regulatory gene near the lac operon. This gene is constantly transcribed and translated to produce repressor protein. The repressor protein binds to the operator DNA at the lac operator, physically preventing RNA polymerase from moving beyond the promoter. When a repressor protein is bound, the genes for lactose catabolism cannot be transcribed into mRNA. When lactose is present in the cell, some lactose molecules are converted to allolactose. Allolactose binds to and inactivates the repressor proteins, preventing them from binding to the operator. The structural genes are transcribed, resulting in the production of the enzymes needed to catabolize lactose. Since allolactose induces or stimulates transcription of the lac operon, it is called an inducer. Notice that the inducer acts by turning off the repressor, not by actively stimulating the genes to be transcribed.
got it?
When lactose is not available, the repressor proteins are active. The structural genes of the lac operon are not transcribed. This control prevents the cell from producing the proteins for lactose catabolism when lactose is not available.
Cool. Make sense now? A little more sense? Let's answer these questions. Let's see. What does the inducer bind to in the lac operon system? So the inducer is the lactose, right? Lactose binds to the repressor, right? Here we go. Second question, I'm going to let you think about this. Which genetic element codes for the repressor protein? So try to think about this carefully. Which element codes for the repressor? The regulatory gene, remember? The promoter region, LAC-Z, LAC-Y, or the operator region? Pause and think about it. Okay? We get one more. When is the regulatory gene transcribed? Always? Only when the regulatory protein is bound to the operator? Only in the presence of lactose? Or only in the absence of lactose? The regulatory gene that is going to make the repressor is always transcribed, right? It's going to bind or not to the operator if you have or not lactose present. Okay, so we did a little exercise here. Now let's go back to where we were. I am not going to talk about repressible operon, so you don't need to worry about the next one. So I'm going to stop here and click on the next video where we're going to talk about mutations and the horizontal type of transfers. Okay? See you soon. Bye.